hear me in the back row there? All right. Can you hear me in the back row there? Okay. All right. Then let's get this as a slideshow. Before we actually start talking about Chapter 3, do people have any questions or comments about Chapter 2? Someone's already pointed out a typo in the Chapter 2 handout, proving yet again that the human eye can miss things if it expects to see something already. It should, in page, on slide number 2-24, there should be pKa in both cases. So the larger the pKa, the less it dissociates. The smaller the pKa, the more it dissociates, and so on. Come on. Okay, there we go. Any other questions about Chapter 2 that people want to ask? Okay, slide 2-24. They should both be down at the bottom of the slide. It's larger pKa, not just Ka. Well, there's two lines there that says, the very bottom line says smaller pKa, that's correct. The line above it says the larger Ka, that should be larger pKa also. Okay. Any content-based questions about Chapter 2? Okay. As far as I know, all three of the previous lectures should be available to you online, either in the context of Icon, where you can view it through your browser, or through Google Video. Has anybody tried to view the lectures and has not been able to make one of those work? Good. If anybody finds a translator for MSS2 into Mac, please let me know, and we can make that simpler. Closer to the time of the exam, we will make available to you practice exams from previous years and review questions. I know it seems very far away already, but if you need accommodations to take a test, please get in touch with me in the next few days or within the next week so we can make sure that those arrangements are made sufficiently in advance that they can be smooth for everybody. Okay? I'm quite happy to make accommodations, but I can't read minds. I keep hoping. Okay. So Chapter 3 is going to be about DNA and other nucleic acids, but principally DNA. And the reason we're interested in this class of molecules is because DNA is the genetic material for all cells and many viruses. And in the course of going through the chapter, we're going to be looking at the building blocks that are used to make DNA. We're going to see how the structure of DNA is a consequence of those building blocks and what those structures, actually, plural, because there's more than one structure to DNA. We're going to take a look at RNA a bit because it's also a nucleic acid and it raises slightly different issues, although there's a lot of overlap. We'll be talking a little bit about the fact that as a molecule it can be melted and so on. We'll review the concept of information flow amongst these molecules and then maybe talk a bit about kind of what the new tricks are that are possible now that we understand more about the structure of DNA and of the behavior of cells. This slide here is just showing you a comparison of two tissues. So this is a light microscopic picture of a section of lung tissue, and you can see this epithelial layer here with beautiful, cute little cilia there that are helping to clean garbage out of the lungs. And this is showing you, by contrast, a cystic fibrosis lung with essentially no cilia there, which is one of the reasons why they have difficulty clearing their lungs, and obviously disorganization in the structure of the tissue as well. The difference between this and this is a single nucleotide in most people. One nucleotide has phenotypic consequences throughout the person. And this is only one of a number of tissues that can be shown to be different in the case of cystic fibrosis. So DNA is very powerful stuff. It just takes a little bit of information to make a lot of difference. I'd like to give a little bit of history to give you a sense of the tempo of these things, how long we've known them, and how they were untangled. Any understanding of genetic material probably is most productively begun with Mendel, who was an Augustinian monk in the 19th century. And he was trying to show or to test Lamarck's ideas about inheritance by planting seeds from one visible type of plant near a related strain that was visibly different but in a different place, and was able to show that it was not the environment that evoked that appearance, but it was something transmissible from the parents. And he did the beautiful experiments that untangled the ideas of dominant, recessive, and so forth, the Mendelian genetics. So he pinpointed, or defined actually, genes as the unit of inheritance in 1865. Almost at the same time, Friedrich Miescher was the first to purify nucleic acids. He did so from pus. If you've ever had an infected wound, the stuff that's infecting that is almost pure lymphocytes. And lymphocytes are 50% or so nucleic acids. And so he was able to isolate nucleic acids from them. The methods that were used at that time made nucleic acids look like very boring molecules because you didn't get intact ones. But that improved with time. Just around the turn of the previous century, people were able to demonstrate that where the genes actually are is as physical structures inside chromosomes. So that was a further clue that something about the content or the structure of chromosomes would eventually explain inheritance. Beautiful experiments by O.T. Avery were able to demonstrate that the genetic material was specifically DNA. They knew at that time that there was both DNA and protein and even some RNA in chromosomes. Avery was able to show that it was specifically DNA that transmitted the trait. He studied pneumococcus, the organism that causes bacterial pneumonia, and was able to show the transmission of being infectious or not through the transmission of DNA. Very beautiful experiments, and if you want sort of a window into the best science of that time, his articles are wonderful. Another couple of scientists called Hershey and Chase were able to demonstrate the same thing through bacteriophage, which are viruses that infect bacteria. This was a helpful cross-check because the methods that Avery had used on bacteria, in some people's minds, still left a little bit of question that maybe there was a little bit of something else besides DNA, and they sort of, Hershey and Chase, basically nailed that coffin shut. Almost the same time, though, a couple years later, a man named Erwin Chargaff made a very interesting observation about the composition of nucleic acids. The methods of analysis were getting better and better, and he noticed that in organisms, the exact composition of the nucleic acids might vary quite a lot, but that there were rules about it so that however much adenine the organisms had, they'd have that much thymine. And however much guanine they had, they'd have that much cytosine. So you could see cells that had, or organisms that had 60% GC down to maybe 30% GC, but the G and C were always equal, the A and T were always equal. Sort of a numerological whisper that something's going on. In that same year, but a little bit later in the year, the actual structure of DNA was uncovered, and it was the combined efforts of several different scientists that were able to do this. A woman named Rosalind Franklin identified the structure as a double helix, and we'll talk a little bit about what she found out about that helix. Francis Crick and James Watson put together Chargaff's rules with her helix to have the idea that there's base pairing going on to account for the helical structure. And so what do chromosomes, these packets of inheritance, look like? And I think what's dramatic is how much more we can see about chromosomes now than we could before. In this picture, when you're looking at the chromosomes from amniocentesis, what has been done here is to use staining methods that can highlight more and less active parts of the chromosome. And so this is a method that allows you actually to get a little window into which ones are being turned on or turned off at a given time. They obviously also make it quite a lot easier to tell that this chromosome is different from that chromosome and so on. But our methods have gotten more sneaky than that. It's possible to use sorting machines to get the different human chromosomes by size. So you can have a pot here of chromosome 1 and a pot here of chromosome 23, and make probes that are specific for either chromosome 1 or chromosome 23. And when those probes are used, you can get what is sometimes called painted chromosomes. And what you can see here, I hope, is that the various pairs of chromosomes are distinctive colors. So anywhere from pale blue to deep red and so on. And this is possible because of this ability to make chromosome-specific probes. And when you paint the chromosomes that way, you can see that in this individual, there is a bit of rearrangement that's gone on, that these two chromosomes appear to have picked up some from one of those guys, and that could be pinned down more tightly with further analysis. But this ability to literally paint chromosomes and see where the structures are is a remarkable accomplishment. The Hershey-Chase experiment was elegant in its simplicity. This little beastie is not the moonlander, but rather it is a virus that is specific for bacteria. It's called bacteriophage because if a bacterium is infected with it, it eventually melts. And so bacteriophage means the bacterium eater. And there are phages that are specific for many different strains. Whole other story off there. And the insight that Hershey and Chase had was to label this virus with radioactive phosphorus and radioactive sulfur. And what was clever about that is that the phosphorus goes uniquely into the DNA, which is packed in here inside the head, and the sulfur goes uniquely into the protein. So the rest of the bacterium is all made out of protein and would pick up sulfur. So now you can track the location of those components by radioactivity. The idea here was that they would infect bacteria for different periods of time, interrupt the infection, and see where it went. The trick for interrupting the infection was kind of cute. They would mix them, and then at 5 seconds or 15 seconds later, they'd hit a blender and blend them apart. The shear of the blender brushed off any bacteriophage that hadn't actually penetrated at that point. And what they found was at that point, the radioactive phosphorus was inside the bacteria, and if they had that, then they were infected by other criteria. So this is a very strong proof of the composition of DNA as the genetic material, protein as its product, ultimately. So what is DNA made out of that it can have such magical properties? There are altogether 5 bases for nucleic acids in general. There are 2 that are called purines, adenine and guanine. There are 3 that are pyrimidines, cytosine, uracil, and thymine. Uracil would be present in the RNA, thymine in the DNA. And the easy way to remember where those are is that it's the AT versus GC ratios. So AT, the T is in the DNA. So these are the bases. You can see that they all are heterocycles. They all have some nitrogen in their rings. These are smaller. Those 2 are larger, and we'll look at that a little bit. If you put a base together with a sugar, and the sugar for DNA would be deoxyribose, or ribose to make RNA, you have a nucleoside. So you can see os for the sugar. And these are both nucleosides. And here you can see a couple of the alcohols hanging off of the sugar ring. This is the one that's going to be gone in DNA. And we're going to be looking at that in a little bit more detail in a second. If you take a nucleoside and add phosphorus to it, you then have a nucleotide. And again, I've mentioned before that the CD in the book is very helpful. I really do commend it to you as a way to get a good feeling for these, because it has very beautiful representations of those structures that let you interact with them and get a good sense of it. Now, the reason they're called what they're called is because adenine and guanine, for instance, can be seen as or thought of as substituted forms of purine. And this is the purine ring. It's a 2 fused rings, a 5 and a 6-membered ring with a lot of nitrogens in it. Guanine elaborates on that by adding an oxygen at the 6 position and an amine group at the 2 position. an adenine modifies it by adding an amine group at the 6th position. So there's 2 additions to the guanine, 1 to adenine. You notice there are numbers in here. This allows us to talk about the positions within the rings from 1 through 6 in the first ring, 7, 8, 9 in the other ring. Obviously those are 5 and 4. So that lets us talk about specific positions if we need to. We can think of cytosine and thymine as substituted pyrimidine. And here is pyrimidine. It's still a heterocycle of the 2 nitrogens, often used as a solvent for certain things, which is kind of interesting. And again, what they've done is to add either an oxygen here for the cytosine and thymine, second oxygen in the thymine. It's a amine group in the cytosine. Uracil and thymine, of course, are these kind of Siamese twins or doppelgangers that can take the similar role in the different nucleic acids. And if you look at their structure, you can see that they both have the carbonyl oxygen, they both have this carbonyl oxygen, and the only difference between the 2 groups is that you have a methyl group hanging off of the thymine. So that's the difference, just one little methyl group to distinguish those 2. Now, nucleosides are those bases that have now been put onto sugars. And here we can just generalize the base as a circle. That's to make it all fit on one page. We mentioned that sugars can be either linear or ring forms. When we're talking about nucleosides, we're talking about specifically the ring form. And now the numbers have primes on them because we already used a bunch of numbers on the bases. So the primed numbers are the carbons in the sugars. And ribose is a 5-carbon sugar that has hydroxyls in both of those places. Deoxyribose is specifically has lost its oxygen at the 2 position. And that's the distinction between the deoxyribose of DNA and the plain ribose of RNA. Any questions so far? Okay. I'm trying to sort of take this carefully because it's good to be oriented. These things make a lot more sense if you're oriented to the whole structure overall. So we can take those nucleosides and now start adding phosphates. So you have either a monophosphate or a diphosphate or a triphosphate. When you take cytosine, it turns into cytidine. Not quite sure why they changed the nomenclature at that point, but there it is. But you can see here the pyrimidine ring structure with its modifications. Here's those purine rings. Okay. So these are all ribonucleotides. So you notice these all have their oxygen there. Those are all ribonucleotides. So adenosine monophosphate is a ribonucleotide. So on. So now what we have is we have something that's extremely hot at one end with a bunch of phosphate groups. We have a sugar in the middle and then the rings hanging off the other side, which are kind of hydrophobic compared to everything else. So this is a very kind of schizophrenic. Let me go back. So this is, if you think about it, this is really a very kind of amphiphilic almost molecule because this end is so polar. And this is relatively nonpolar. It's not strictly nonpolar, but it's certainly less polar than the other 2. So we have a molecule that has a tendency to have a difference across it. To take those nucleotides and turn them into a nucleic acid, bonds have to be made between the riboses. And the way the bonds are going to run is there's going to be a phosphodiester bond. So this is a phosphate group that has an ester linkage to this sugar and an ester linkage to that sugar. So that's why it's a phosphodiester bond. And the specific places it links is from the 3' hydroxyl in the ribose ring to the 5' hydroxyl that was sort of sticking up in the air. Let me just back up one again. You notice here you notice in the nucleoside that the 5th carbon of the ribose ring is sticking up in the air, so to speak. That's the carbon, that's the oxygen that's going to participate in the phosphodiester bond. So if we have this now, we have a phosphodiester bond that links always the 3' carbon of one to the 5' carbon of the next one, and so on down the line. What that does is it makes a molecule that is polar. That is, one end is different from the other. This end will have a free 5' end with a phosphate group sticking off. This end has a free 3' end with just a hydroxyl sticking off. And so the molecule is not symmetric left to right. It's a very asymmetric molecule. We talk about here as a residue. This is a thymine residue, a guanine residue. It's what's left of the triphosphates. Is this molecule RNA or DNA? Right. So if you look there, the 2' position, just a hydrogen, and so this is the DNA. Okay. Any questions about this before we move on? Okay. This having a good sense of the structure of the backbone will help you in many contexts, believe me. Okay. Now, it was this insight of Watson and Crick that the structures of the bases could be used as a way to assemble DNA into paired structures. And the idea here is that because adenine has the capacity to make hydrogen bonds, and so does thymine, guanine, and so forth, the packing of them is such that an adenine-thymine pair takes up about the same space as a guanine-cytosine pair. So you can see, you can almost in your mind superpose those two pairs. The AT pair has only 2 hydrogen bonds here, whereas the GC pair has 3 hydrogen bonds. And you can see again the different kinds of hydrogen bonds we've talked about. Here's one between a hydrogen and an oxygen. Here's one between a nitrogen and a hydrogen, and so on. So most of the kinds of hydrogen bonds we've talked about are going to be visible in the bases of DNA. It's always a purine to a pyrimidine. That keeps the space, the sizing, the overall footprint the same. The other thing to notice about this is there are still potential hydrogen bonding sites left on these structures. So here, for instance, that could obviously take part in hydrogen bonds. That could take part in hydrogen bonds, and so forth. So even in the double helix, there are unoccupied sites that could participate in hydrogen bonds, and that's going to turn out to be important later on. But what does that look like then? All we've talked about so far is chemistry. It doesn't talk about structure. We showed you the phosphodiester bonds and the fact that it's a long, skinny molecule. What shape does that long, skinny molecule take? Depends on using other methods. And the method that was most productive at the beginning was X-ray crystallography in the hands of a woman called Rosalind Franklin. And she had learned on amorphous carbons how to handle tricky samples and had some handles for approaching this. At any rate, she was able to determine a number of important things about the structure of DNA. First of all, that it was a double helix, that there were 2 strands present, that it was anti-parallel. What that means is that the 5' end of this one is going down. Its partner will be a 3' end. That didn't have to be true. It turned out to be the way it's true. And so what that means is they're anti-parallel, that the one on this side starts from the 5 and ends with a 3. The one on the opposite side happens to start with a 3, end with a 5. Could be either side. It's not magical left to right, but there's always a 3' and a 5' end talking to each other, and so on. She was also able to tell on the basis of the X-ray crystallography that the bases were going to be on the inside and that the phosphates were on the outside. She could detect that it was a right-handed helix. So if you kind of look carefully, you can see that this is rising to the right. Every helix either rises to the right or rises to the left. So you can characterize a helix as having a handedness. And one way you can get a feeling for that is if you look at the pedestrian bridges on Riverside the next time you go by. One of those bridges has a right-handed helix on its ramp, pedestrian ramp, and one of them has a left-handed helix on its pedestrian ramp. So take a look at them and see if you can get a sense for them. She was able to tell by X-ray crystallography that the pitch, the distance for one complete repeat was 34 angstroms, and that the distance across was 20 angstroms. So she was able to give an overall general shape to the double helix, the DNA molecule. The insight that Watson Crick had was that by using Chargaff's rules and by using hydrogen bonding, you could get a smooth packing inside the helix so that all the bases could tuck in there nicely. They noticed the parallel character of these bases. If you look at them, they're fairly close to parallel to each other. And their model also brought out something that wasn't visible by the crystallography possible at that time, which is there's a major groove. Here's a wide space in the helix. So that's a major groove of the molecule, and a narrow space called the minor groove. And that comes out of that. Any questions about this structure, which is, without question, one of the most beautiful structures in the world? Okay, here, I can keep moving there. It's an intricate structure, and it can be hard to see all of it at once. Different approaches are taken for representing it depending on what you want to get out of it, and this is just 3 of them. If what you want to emphasize is the phosphate backbone, this is a good representation, where it's using colors for the atoms, so you can notice the ribose sugars and the phosphate in the phosphodiester bonds. I think this brings out very clearly how in this structure of DNA, that phosphate backbone is right there on the outside, and that it's helical. I think the helical character may be a little easier to see on this one. Sometimes it's more worthwhile to look at the bases, and here what they've done is to color each of the bases with its own color, so you can see the complementarity, that you're only getting blue with yellow and you're only getting red with green and so on. Both of these give you a misleading sense of the molecule because they look so empty. The space-filling diagram like this is, without question, the easiest to see the contours of it. You can see that there really is a major groove here, and there really is a minor groove there. It's much easier to see those grooves in a space-filling diagram. They were present over here, it's just easier to see it here. It may also be easier to see that it's a right-handed helix. You can see that backbone going up. What's not very easy to see is how those things are organized inside of it. And again, the CD has some wonderful pictures where you can, switch between the different representations and rotate it around in space and so on. Now, so far we've been talking about DNA folding, but RNA can fold also. RNA is, again, an asymmetric molecule with a 5' end and a 3' end. It's a phosphodiester backbone. In nature, though, more often we see single-stranded RNA molecules than double-stranded RNA. Double-stranded RNA is more common in a virus than anywhere in the cell. Most of the RNA in cells, bacteria or eukaryotes, will be single-stranded. But it's still possible for it to form helical structures. It just doesn't form bare ones. And so what you can see here, this is a structural diagram for a transfer RNA molecule. And what I think you can see from this is that there's a helical structure here. So this is a part of a helix, and there's some helical structure in there, and so on. So it's possible in RNA to base pair within one strand. Instead of having a 2-stranded anti-parallel structure like DNA most commonly is, RNA is one that kind of tangles onto itself. One of the consequences of this is that RNA has a remarkable variety of structures compared to DNA. Structures in the sense of the overall 3-dimensional packing and organization of the thing. And a consequence of that is RNA has a wider variety of kinds of functions. The kinds of things they do are more various than what DNA does. DNA is kind of a boring molecule in that respect. It can make more DNA, it can make RNA. That's sort of what it does. RNA, on the other hand, can take part in a lot of different functions. Okay. And DNA and RNA can hybridize. They both have the bases, and the T and the U are equivalent for base pairing purposes. And so you can make a base pairs between RNA and DNA. And if you do that, it has a rather different shape to it. You can see that it's a flatter structure. It's wider. I think you can see here that the bases are more tipped relative to the axis of the helix. In the DNA double helix, the bases were basically flat. And this actually looks like what's called the A form of DNA. We're going to see a little bit more about that in a bit. All of what I talked about up until now in DNA was the B form. And we're going to see why there's differences between B and A in a second. RNA can make double helices, and in that case, its structure looks more like the B form. Most common place to find double-stranded RNA molecules that are two separate strands would be in viruses. This is showing you some of that variety now of the different overall configurations that DNA can take on. Almost all of what we've talked about so far, we've been talking about what's called the bDNA form here. I think you can see here that this representation is the same as this representation. You've got the major groove here and a minor groove there, major groove here, minor groove there. But that isn't the only configuration that DNA can take on. The A form here is quite visibly wider. It's shorter for the same number of nucleotides. And you can see that the distribution of the phosphates is still on the outside as it was in the case of the B form. The A form is commonly found when there's less water around, but there are other things that can trigger the A form as well. And then there's zDNA. zDNA gets its name because the backbone zigzags up as you look. It sort of goes jing, 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 back and forth. It's a left-handed helix. It was initially found by people who were looking at DNA's a very odd composition. And then somebody went to look at DNA from organisms and found patches of that odd composition in real DNA. So it's emerging that zDNA is a configuration that DNA can take on in living cells for very specific purposes. So although the great majority of the DNA in a living thing will be in the B form, both of these forms, and especially the zDNA, are other possible configurations that the DNA can take on for specific purposes. An odd thing about the zDNA here is that it's sort of an asymmetric helix. You can see most of the phosphate is sort of pushed off to one side, and the bases are kind of off-center as well. Actually a little hole in aDNA if you look carefully. And this is then summarizing those things. Again, I don't expect you to memorize the numbers. This is a way of having the specifics of how these different forms differ. The two right-handed ones here, this is showing you the residues per turn, that bDNA has exactly 10. And there's got to be an error there. Oh, that's okay. Okay, there's 11 residues per turn in aDNA and 12 in zDNA. So they have different tightness of twisting. And they also differ in how much they rise. So aDNA is squatter, zDNA is more elongated, and so on. The base pair tilting, you can see that in bDNA it's nearly flat. In aDNA, which is like the DNA-RNA hybrid, kind of tilted to it, and so on. So very interesting to see how much the same molecule can differ in its overall structure, depending either upon the environment or the composition, the sequence composition of the molecule. Any questions about that before we start to talk about DNA melting and annealing? No? Okay. So we've talked a little bit about how there's hydrogen bonds between the bases, and that there's 2 between the AT pairs, 3 in the GC pairs. One of the consequences of that is that it gives very smooth packing, that the interior of the helix is quite full. If you remember back to the space filling diagrams, that you have kind of a completely solid interior, if you like. There's no extra space in it. The forces driving that, though, are not the hydrogen bonding. Hydrogen bonding is what accounts for how it packs, that it packs with this strand and not that strand, that it is in register and not out of register. But the hydrogen bonding is not what drives the folding or the unfolding of DNA. And the reason for that, if you think about it, is that in the unfolded state, the various sites that are hydrogen bonded inside the DNA molecule are still hydrogen bonded. They're just hydrogen bonded to water molecules. And so the assembly of the DNA double helix doesn't really make any energetic difference to those hydrogen bonds. So the force that drives it from unfolded to folded is not due to the hydrogen bonding. That just accounts for where the spacing goes. It's due to 2 interactions called stacking interactions. You remember that all of the bases have these aromatic rings, and there's interactions between the clouds of electrons up and down called stacking interaction. And so there's an actual positive interaction between the bases. There's also some contribution from the hydrophobic effect. That is, the base part of the nucleic acids is less hydrophilic than the rest of the molecule, and so there is some tendency for the hydrophobic effect to drive it out of the solution. I've been trying to find a good statement of the relative contribution of these two things, but I'm not sure I believe any of them yet. There definitely are both of them contributing, and the stacking interactions are unique, basically, to the nucleic acids in living systems. The GC pairs have 3 hydrogen bonds, and as a consequence, they are more paired to each other, and they also pack a little tighter. And so if you have DNA that has a lot of GC content, it tends to be more stable against being melted by heat. And we can see this in things like the thermophilic organisms. So if you isolate something out of a hot spring in Yosemite, it might have 70% GC in terms of making up its DNA. This is kind of cute because we're looking down the axis now. That wasn't a view that we had a lot of access to before. And you can see here how in the interior, the bases really occupy all the spaces in there. Okay. I mentioned already the idea of melting without actually specifying what I mean by that. And it's something like this, that if you think of the normal state or the biological state of a DNA molecule as the double helix, you can represent that schematically like that. Now, the double helix is not a covalent structure. It's only held together by non-covalent interactions. And so as you increase the heat in the solution, at some point you're going to end up with a mixture of free single strands of nucleic acids because the banging around of molecules will just end up ripping it apart and it'll stay apart. But it's not an all or nothing thing at the transition. It's not that they're all together first and then they're all apart. Instead, what we see is something that you're going to see again in many, many different contexts, which is that there is a transition between these two states that has a curved character to it. Sometimes this is called a sigmoid curve. And so very little of it has melted here, and as temperature rises, more and more of it melts, and then finally we get to some point where all the molecules have separated from each other. So at this point we can say it's all melted. At this point we can say it's all unmelted. And in between there's a population that's melted and a population that's not. We could ask, I mean, that's a broad range. You see for this particular diagram, they're showing you close to a 50-degree spread, and that's quite possible for some DNA molecules. So it'd be nice to have a better way of talking about that, and the approach that's been taken and is used for a great many things is to ask, at what temperature are half the molecules melted and half of them intact? And we call that TM, the melting temperature, not because there's no melting before that temperature, but because at that temperature it's half and half. This idea of picking a value at which it's half X and half Y is going to recur in many contexts. It's a biological strategy because biological systems display this kind of behavior very often. One of the interesting things that comes out of this is that the exact TM, whether it's a lower or a higher one, is a measure of the stability of the molecule. And so if you have a lot of GC content, you'll have a higher melting temperature. If you have a lot of repetitive sequences, you'll have a higher melting temperature. So in some species, there might be 90% of the DNA is repetitive sequences that don't contain genes. That kind of sequence will melt at a higher temperature because there's so many of them there. You might have noticed, or maybe you didn't notice, what the vertical axis is. My axes are all labeled, unlike some ecology axes. And in this case, it's absorbance at 260 nanometers. And what is that? Well, it's that if you shine light through a solution of nucleic acids, that the bases either do or don't interact with that light, depending on whether they're stacked or not. That if the molecules are stacked up in a double helix, they're not very free to interact with that light. They don't absorb very much of it. Whereas once they're free and floating around, those bases are out and they can act like little sunglasses and catch the 260. And so the reason you see more absorbance is because the melted molecules are free to absorb the light. Okay. Let me just see. Let me just take a second here. Okay. This is showing you that in a little bit more sort of diagrammatically, but also showing you that it's, again, not an all or nothing thing. So if we were to start with a well-paired double helix and heat it until all the molecules were melted, and what they've done here is to try to show you that the different bases are colored different ways. If you were to cool it rapidly, so you have this tube that's full of melted DNA and you put it into an ice bucket, the change in temperature would happen so much more quickly than the normal folding that many of the molecules in it would be mismatched. They would have some base pairs that are correct. You can see here a yellow-blue pair. You can see some that are inside the molecule and so forth. So you can have denatured, these are mismatched sites. But this is a reversible process. This is not something that harms the DNA in any way. If you take this mixture of badly paired nucleic acids and you warm it up, but not to melt it, just they're suggesting here in the range of like 20 degrees below the TM, and we'll go back and see. You remember here's the TM where half of them are melted and half of them aren't. What they're suggesting here is that by going down in here, where just a few molecules are melting, that what you can do is every once in a while, one of these mismatched ones will melt, and when they reform the correct one. Because you can easily imagine that this structure has a lower melting temperature than this structure. So by warming it up, not to the TM, but below the TM, you're going to allow a gradual renaturation of this mismatched helix up into that. And this is quite reversible. It can be done dozens of times. The molecules have no memory for where they've been. This turns out to be an important property if we want to do various analyses of how molecules work, and we'll be talking a little bit more about that in a little bit. Okay. Now, it's this ability to reanneal, this ability to find a specific sequence and do that in a very specific way, can be used in a wide variety of ways. And this particular one is showing you the idea that you can selectively identify particular sequences in a large mixture of things. So if we start by having some samples that we want to analyze in some way, and what they've done here is to show you immobilized nucleic acids. This matrix might be something like phosphocellulose. It might be something that's a commercially proprietary thing that holds on to the nucleic acids, but in a way that they can still interact and renature and denature. Up here what we have are samples that are going to be used as probes. They are single strands. So these are presumably strands that will be complementary to these strands. If these strands are radioactive, we can then mix them with our immobilized strands. And then we have a mixture in here where you've got radioactivity in all kinds of places, both on the nucleic acids and elsewhere. But the binding of the probes to the nucleic acids will be stronger than the other ones, and so you can wash that off. And then you'll find that one particular probe will bind to one particular sequence, and that will show up as radioactivity in just the one place. This process of washing things away you can control in a variety of ways. Temperature is one obvious one because we've talked about TM already. Another one is salt concentration. You can think about that backbone of the phosphates. You can imagine there's a lot of charge density there, and so the salt concentration can increase or decrease the ease with which those double helices can form. And there are other conditions as well that can be used to control this. This particular one is being shown as a radioactive probe. It can also be done with fluorescence now, so you could have potentially a different color for each of those probes and be able to probe more than one thing at once. Why don't we call that a good quit? This is a nice place to stop it.